Okay, can you see that?
Yep.
Okay, awesome. So I'll go ahead. So I'll be talking about a project that I worked on for my PhD, looking at the seasonal activity, spatial distribution, and physiological limits in subterranean termites. So resource partitioning is a major question in ecology. Gauss's law states that two organisms cannot not occupy the same niche without outcompeting the other. So in other words, there can't be two things doing the same thing in the same place. This is particularly exemplified in social insects, where we have colony recruitment. And instead of just having individuals finding and using resources, there's an entire colony that can be recruited very quickly. In social insects, resources are generally partitioned through competition, behavioral differences, or use under different climatic conditions. For this, I'm focusing on subterranean termites, on the genus Reticulotermes. Many of these species occur often in symmetry. I'll be focusing on these three widespread eastern species, Reticulotermes flavipes, hageni, and virginicus. And as you can see, there's a very wide area where all three species occur. And when you narrow into an individual field site or even an individual resource, you can often find multiple, or you can at times find multiple species in a single log. So these species are hypothesized to partition these resources by selecting different log sizes. There's some data, or by foraging under different climatic conditions. So these different climatic factors, a previous study, Hausmann et al. In 2001, found the foraging preference for temperature and moisture when comparing just the two species, flavipes and hageni. They found that flavipes was more active in these cool, wet months, and hageni was more active in the hot, dry months. So it kind of flipped at different times of the year. Additionally, in laboratory studies, Reticulotermes flavipes was found to be able to withstand flooding for a longer period of time than virginicus. But my objective for this study was to understand how this resource partitioning enables all three of these species to coexist on the same resources, as many studies had looked only at individual pairs, and I wanted to have a better view of all of these species. Specifically, I compared the species-specific foraging behavior for these three species, assess the impact of soil temperature and soil moisture on the foraging activity, and then test if these physiological limits, as I was talking about previous studies have investigated, if these physiological limits can predict the impact that climate has on these foraging patterns. So to do this, I set up a field site in Eastern Texas at Sam Houston State University Center for Biological Field Studies. This was a nice pine wood forest. There was lots of downed wood, and almost any log you rolled over had plenty of termites in there. In this site, I set up two plots that contained 14 by 14 grids of pine wood stakes spaced 2 meters apart. They were about 240 meters apart and separated by a river. So it was unlikely that these foragers were from the same colony at these different sites. I measured the soil temperature and soil moisture every 2 minutes using hobo data loggers and collected samples for the duration of the study, which ran for 28 months. For the first 2 years, I identified them genetically to species and characterized the foraging areas of the different colonies for just the first 6 months due to sample size constraints. I also looked at the foraging duration. So this is an example of what the kind of the corner of my data sheet looked like. So for this, each of the rows represents a monitoring station, like 2113A. Each of the columns is a month. And so when there's the different species are represented by different colors and their initials. So in this first row, there's 2 consecutive months of Reticulotermes hageni foraging, which I would say is a duration of 2 months foraging for this colony. The next row has 5 flavipes months in a row. And so that would be a duration of Reticulotermes flavipes foraging for 5 months. In the first 6 months, we didn't see colony turnover of the same species in the same monitor. So for the duration of the 2-year experiment, we assumed that if it was the same species in a monitor in consecutive months, it was the same colony. So looking at the results, as you would expect, this is the graph showing the variability of the monitors over the duration of the study. So on the x-axis is the months. So you can see it starts in September of 2016 and ends at the end of 2018. These red dots are the percent of active monitors. So because as time moved on, I added more monitors to the area. And so I'm using percent active monitors to show how the foraging activity is. These vertical lines are the end of the year, so the end of January 1st. And so you can see in the summertime, these increase, and in the winter, these decrease, as you would expect. So there's a strong correlation positively to temperature and negatively to moisture. So the temperature, the green, and the moisture are the black dots there. And so you can see that generally the winters are wetter and cooler, and there's lower foraging. There's also some sharp declines. You can see the first sharp declines when Hurricane Harvey came through, and it knocked off the foraging considerably. When these different flood events occurred with this high moisture, there would often be standing water in the plot. And when you take the stakes out of the ground, there would be standing water in the hole that would fill up where the stake was. When looking at the species composition for the duration of the study, you can see that the plot was dominated by Reticulotermes flavipes. That was a majority of the stations, followed by Hageni, and then Virginicus was more rare. For the first 6 months, we characterized the colony foraging areas. So these figures show us where these colonies foraged for the entire duration of the 6 months. So these different shapes represent all of the areas that this colony foraged. The red lines separate colonies that were very close, and the different shapes are the different colonies. So we had about 32 flavipes colonies, 16 Hageni colonies, and only a single Virginicus in one of the plots. And as you can see, as was found in previous studies, the species have overlapping foraging ranges. So there's many occurrences where the same monitoring station in the same area is inhabited by different species at different time. However, the colonies of the same species do not overlap. So you can see these yellow shapes, particularly since flavipes is more abundant, the yellow shapes never overlap with each other. When looking at the foraging duration by species, so again, how many consecutive months they foraged is the foraging duration. You can see that flavipes foraged for a longer duration than the other species, averaging about 3 months of foraging duration, whereas the other species were only about 1 month and then gone. Interestingly, there were occurrences where a monitor would be occupied by 2 different species in consecutive months. So there would be, in 1 month, flavipes would be there, and in the next month, Virginicus. And while there were about 160 of these occurrences, and flavipes had the most, there were also the most flavipe samples. So when scaled by occurrences, Reticulotermes virginicus initiated foraging in a monitor that was already occupied about a quarter of the time. So a quarter of the time that it started foraging, it was foraging in a station that was previously foraged in by another species. So this is an interesting strategy that Virginicus might use to find resources. When looking at the impact of temperature and moisture on species foraging, these are the correlations again. So generally temperature was positively correlated and soil was negatively correlated to, in this case, discovery rate, or how many stations they're discovering each month. So for discovery rate, when doing a multivariate analysis, we found that both temperature and moisture contributed to this correlation, whereas Hageni was mainly influenced by temperature and Virginicus mainly by moisture. When looking then at the leaving rate, or how likely they were to leave a station in that month, Reticulotermes flavipes was not significantly correlated to either temperature or moisture. Hageni was correlated to both temperature and moisture, and Virginicus was only correlated to moisture. So these different climatic factors are shaping these foraging patterns. We then wanted to see if we could find some of these innate physiological abilities in the lab that could help describe what we saw in the field. So for this, we measured a few different variables. We looked at the upper lethal limit, or the maximum temperature an individual could withstand. For this, we exposed individuals on the heat block, separated in separate tubes. We started this at 33 degrees Celsius, increased 1 degree Celsius every 5 minutes, and examined them after 5 minutes to see if there was any visible movement, and then they were declared dead. We did a desiccation assay where we put groups of termites in an empty petri dish and exposed them in a desiccation chamber, assessing mortality for these different colonies of the different species every 3 hours. And similar to previous studies, we did the submersion assay, where we forced groups of termites underwater for variable amounts of time, up to about 36 hours. We then removed them, put them in a petri dish with filter paper, and assessed their mortality 24 hours later to see if they were able to survive. And surprisingly for me, there were termites that survived 36 hours of complete submersion. So to look at these differences, for the upper lethal limit, we have here our field site in Central Texas, but we also added a field site up in Arkansas to compare these 3 species. And while the difference, or the upper lethal limit, is different between Arkansas and Central Texas, the Central Texas ones were able to withstand more heat. There was no difference of the 3 different species within a site. So these species have the same tolerance when exposed to the same conditions. Similarly, for the desiccation assay, all the different colonies, this is 2 or 3 colonies of each species, were able to withstand desiccation to the same extent. So there was about 24 hours was the lethal time that 50% died, and there was no difference by species. Same thing for submersion. There's a lethal time 50 of about 23 hours and no difference by species. So overall, we found that these species may coexist by foraging under different climatic conditions. So we observed that Flavipes continued foraging after these high moisture events was not influenced by these conditions and their leaving rate, and were generally more stable, foraging for a longer duration in the station. Reticulotermes hagenii increased their foraging under high soil moisture, and Virginicus increased foraging under low temperatures. And they also had that strategy of utilizing resources abandoned by other species. Unfortunately, we did not detect differences in the heat, submersion, or desiccation tolerances to try to explain these patterns on a physiological basis. And this is possibly because these species have the ability to avoid these extreme conditions by moving through the soil profile. So because termites are tunneling at different depths, they're able to behaviorally move and avoid these conditions rather than just being able to tolerate these. And these species-specific preferences can extend then to habitat selection. So as we talked about, Flavipes, the leaving rate was not impacted by environmental conditions, and this may enable them to hold on to food resources in times where other species are forced out of their resources. Hagenii, we found forage during wetter periods, and so this may cause these species to select shaded, humid areas to have these preferential resources. And Virginicus foraged was generally influenced more by foraging in cooler periods, and this may enable them to feed on things like deeper tree roots that are going to be at a cooler temperature in the soil. And through this heterogeneity of microhabitats may help reduce the interspecific competition and enable these species to coexist in the same area. For our study, we used these below ground resources. So we inserted the stakes in the ground about 6 inches into the soil, and we looked at the termites that were foraging through the soil. But this also would imply that there might be resource selection for these above ground resources, because above ground resources are going to have a lot more variability without the buffer of the soil. So we found that Reticulotermes virginicus is known to prefer larger resources. Studies have found this in larger logs, while hagenii is generally found in smaller logs. And this relates to the fact that the Virginicus in these larger resources is buffered from this moisture variation because they're in a larger log, whereas hagenii prefers smaller resources and can withstand this variation. Unfortunately, again, the opposite pattern occurs for temperature, where Virginicus is able to withstand more variation in temperature, and hagenii is the opposite. So this may help to explain it in terms of moisture, but temperature may be explained by different variables. We also can consider nest relocation. So Reticulotermes have these amorphous colonies that are able to move into different areas. There is at least one instance where I pulled up a stake and found reproductives in the stake that had been moved in as they hadn't set up their entire colony there. So these colonies are able to switch between food resources, although they're generally remaining in these well-defined areas. And so additionally, when an enemy is encountered, they can either relocate from that resource or remain in fight. And for this study, I was not looking at a fine enough scale to see these individual interactions. However, we see kind of the aftermath of one colony replacing another in a stake. And so in this, because we saw Virginicus took over more colonies, it may be a better competitor. And this makes sense because these large widespread colonies of Virginicus are more likely to interact with other species rather than other colonies of the same species. So these competitive interactions may also be important to how they're partitioning these resources. So overall, we found that there are differential responses to climatic factors in these species, and this may help explain how they're partitioning these resources and existing in the same area, seemingly on the same resources. But additional factors may be important, including the microhabitat heterogeneity, above ground resources, extreme weather events like the hurricanes and other extreme floods that came through, and nest relocation. And with that, there's many people and funding sources to acknowledge that help this, particularly my major advisor, Dr. Ed Vargo at Texas A&M, and many other supportive people in the lab. And I'm happy to take any questions. And if you have more details that you want to see, this paper came out earlier this year in the journal Insects and is open access. So you can go online and grab this, but I'll take any questions if we have time.
Yeah, I think we have time for one question. Would you expect Virginicus to be more likely to hit a termite bait station being fed on by another species after it has been controlled, or other species in the foraging areas?
Yeah, from seeing this kind of interaction, yeah, I would expect that Virginicus would be more likely to hit a bait station that's already been occupied by another colony. In this, I don't know the exact time scale, if the other colony was there when Virginicus took over, or they just took over an abandoned station using those old tunnels. But yeah, I would think that the less disturbance would make it more ideal for Virginicus to hit that station.